Hi, hello everyone. This is Kokulnathan from Taipei, Taiwan. It is my great pleasure to brief you about the importance of my research work and especially about neurotransmitters, dopamines in electrochemical sensing. As today's session or today's briefing will be for in a general topic, so I'm gonna take the complete sessions in the general manner, and preferably I'll go with in English only, so that it can accommodate to large number of audience, and it will be a quite interesting analysis, and it will be quite interesting talk between people. So before going into the topic, I just gonna brief about myself. Thing about me, I was born in Erode in Tamil Nadu. As you all know about, it's a very good place where I have completed my bachelor's in 2012 and master's in 2014. After that, Taipei has given more much of a importance, and it drew my attention. So, where I have flown to Taipei for my doctoral studies, and I have persuaded my completed my doctoral studies in 2018 material science and engineering department under a distinguished professor Shen Ming Shen, who is a well renowned professor and scientist in electrochemical sensing and analysis. Basically, my research interest covers in nanocomposites of rare earth metals, lanthanides, and transition metals, where bimetals and synthesis of nanocomposites, and its enhancement and its determinations in electrochemical analysis, and also about its characteristics analysis and variations using surface Raman acid spectroscopy, and also through in energy conventions and in other aspects. So there are quite number of awards which have been researched in year to year time, which was helped me to career up. So coming to Taipei, Taipei National University of Taipei, National University of Technology (NTUT) is 100 years old, where it was started in 1912 in a beautiful country called Taiwan. Taiwan is usually said to be the heart of Taipei. I need to say, in this chaos, in this pandemic situation, COVID-19, even though Taipei is very closed and Taipei is a Republic of China, it has gained its independence before 110 years and 108. Uh, so, coming to about my home university where I have completed my PhD, it's a 100 years old university which has started in 1912 after the liberation from China. They were a Republic of China, Taiwan, Republic of Taipei, it is called as. And the very good thing about the country is it is heart of Asia, and I just want to say a few words about this. In this pandemic situation, as we all know about COVID-19, COVID-19 is very drastic thing which has hurt the complete human kind. In these pandemic situation also, Taiwan hasn't been closed. Means it was not closed. It was not in a lockdown, nor it was in any sort of restriction. Without any restrictions, like it's being a user friendly, the country is being progressive by controlling the COVID-19 disease. And there are only about 420 cases and only 6 deaths, which is quite interesting. Whereas WHO, everyone should admire through the Taiwan and to should know about these things. So the love to this country, and the importance of this country and the scenic beauties, and especially the 101 World Trade Center, 101 World Trade Center has brought me to Taipei uh, to persuade my post uh, studies. After this post studies were under the guidance of Shen Ming Shen in NTUT, have completed in 2018. So coming about education in Taiwan, India has a very good relation with Taiwan. Even it is said that here Shanghai Sheikh used to have a very good interrelations with Mohan Chan, Gandhi, where they used to participate in Quit India movement. From that time to till now, India and Taipei has a very good interrelations. And coming to education overall, in education in Taiwan is very good. There are quite number of universities, quite number of scholarships also who are interested to persuade in bachelor's, masters, and doctoral studies also in every field. And basically Taiwan concentrates more in technological analysis, nano enhancements, and in medical enhancements also. So the quick facts are like, there are the universities and the grading scales, whereas this completely follows US pattern and US educational systems, where a quite number of scholarships also will be given, like MOF, 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 Taiwan scholarships, and university special scholarships, and even MediaTek scholarships, and CTCI special grant scholarships. There are many scholarships which are being given by both Indian government and Taiwan government, which has been increased, like more number of Indian students and other parts of the country students are also being attracted to be as a part of Taiwan. So that's about the country and things. And coming into the talk today, so coming about the overview of my topic today, the I'll be speaking about what are neurotransmitters, why neurotransmitters is so important in detecting, and how dopamine plays an important role. What's the vital conditions in electrochemical sensing analysis and electrochemical determination of dopamine in live cells to cure Parkinson's diseases or some other various diseases, how the transmission happens, and also its applications and its future. Before going into the topic, I'd like to say about the person who has been behind this. Neurotransmitters was first discovered by an Australian scientist earlier in 1912, where neurotransmitters are basically endogenous chemicals that enables neurotransmissions. The type of chemical messengers that transmit signals across a chemical synthesis from one neuron, from one nerve cell to another target neuron, like muscle cells or gland cells. They're received through neurotransmitter receptors to the target cells. Many neurotransmitters are synthesized from simple plentiful of precursors such as amino acids. There are quite a number of amino acids which are used, which are available, and only requires is a small number of biosynthetic steps for conversion, where the conversion happens and the phenomenon and the metabolism is being chosen and spoken over there, where neurotransmitters are essential to function for various number of complex neural systems. The exact number of unique neurotransmitters is still yet to unknown, uh, but there are more than about 200 which are being identified. So about the discovery in the 20th century, the various scientists has gone through uh, for its importance, and the transferring syndrome left, which has been conducted, and many scientists has come forward to know about the nerves of nerve conditions and various nerve wag analysis. So in brief, as in journal, neurotransmitters are quite number of chemicals made by nerve cells called neurons, which are used to communicate messages across various parts of the brain um, and between the rest of the body. So how a neurotransmitter works? Neurotransmitters works by sending chemical signals between various cells called neurons through brain, spinal cord, and rest of the system. And the categories of it or divides and exclamates between two conditions or three important analysis. So importance of, like before saying about the what is the condition in this. How these are identified? There are main categories for identifying neurotransmitters. The chemicals must be synthesized in the neuron, or otherwise it must be present in this, where the neuron must be active. The chemical must be released and produce a response in quite certain targets, where the response uh, must be obtained when the chemical is experimentally placed onto the target. The experimental analysis should come forward, and the mechanism must exit for removing the chemicals from its sites, and also for the activations after the work is done. There are advances in many advancements which are going being run in pharmacology, genetics, and chemical neuroautonomy systems. Especially for neurotransmitters, it can be applied as they carry messages between neurons via influence of the post-synaptic membranes, which have little or more of the conflict variants and to the structures of the synapses. So, in general, if you say what are the quite few important neurotransmitters, like there are how the structures of the neurotransmitters been like here are quite different number of neurotransmitters which has been used, like adrenaline, noradrenaline, dopamine, GABA, glutamate. So where in this you can see the carbon atoms and the hydrogen atoms which are being combined with oxygen and nitrogen variations, where the rest molecules and the hydrogen atoms varies according to the types and anatomical localizations of neurotransmitters. So particularly there are many types, many different ways to classify neurotransmitters, dividing according to amino acids, peptides, monoamines. So basically, what are amino acids? Amino acids are glutamates, asparates, deserins, and glycines, which we use, and peptides are substance P's, cocaines, optics peptides, and these kinds of things, and monoamines. Basically, I just want to talk about dopamine today. So dopamine comes under the monoamines, which helps in conducting single ions, optoid receptors, and most of these monoamines like dopamine receptor, trace amino acid receptors, and these conditions. So in action, neurons, how this happens. Neurons form elaborate networks through which the nerve impulses and the action analysis variations, where each neuron will be consisting of around 15,000 connections with neighboring neurons. In general, if you say how this happens, like if you take a brain, uh, here are like two main types of neurotransmitters, like how to say, excitatory neurotransmitters and inhibitory neurotransmitters. Excitatory neurotransmitters stimulates nerve activities, while inhibitory transmitters decreases the nerve activity, which leads to mental illness, depressions, and imbalance these certain neurotransmitters. So how this happen? Quite in a brain, if you think about their cerebellum, medulla oblongata, where the neurotransmitters are linked under 15,000 connections, and all these are intercalculated and where every part of this is uh, consisted and calculated. Dopamine. Why dopamine? As I said earlier, dopamine comes under the dopamine comes under monoamines, and that dopamine as a monoamine has a number of functions in the brain, which includes motor behaviors, pleasures that are related to the motivations, and it also emotional arousals, reward systems, and it plays a very crucial roles in reward systems, Parkinson's diseases, uh, Parkinson diseases, and to low levels of dopamine, which leads to help in Parkinson's a lot. So, what is this Parkinson diseases I'm talking about a lot? Parkinson's disease is a long-term degenerative disorders of the central nervous system that mainly affects. It affects mainly the nervous system. Uh, uh, the complete motor system of the nervous system is being affected through Parkinson's. As the disease worsens on, like if it completes onto the body, if it attacks to the body more on that, non-motor symptoms becomes more common, like early in the disease, as it should, there will be early stages, like preliminary studies or preliminary things will be like only shaking, resistance, slow of movement, where thinking behavior problems will occur, dementia starts on, and gradually it leads to depression, and also anxiety levels will go on. Other symptoms include sensories like sleep deprivations, emotional problems. These all sort of consider as Parkinsonian syndromes. So that's as we are not talking about that, and our main motto was today about to think, analyze how, or to understand how dopamine is considered as one of the prominent material for electrochemical analysis, or in especially neurotransmitters. Dopamine uh, is a neurotransmitter that helps usually sends signals to the brain. Uh, as I said earlier, it is connected by controlling the movements and the production of the dopamine paths, or brain can related to Parkinson's. As I said about Parkinson's, the stiffness completely inherits the motor systems of a person, where it leads to the mental illness, and it shakes. So how the signal analysis is done, how a receptor is being done, and this photo you can see clearly uh, how how it is going. It is clearly mentioned when the dopamine is being inhibited into the body, the metabolites and the receptors start acting, where the left is being conjugated and the signal started to respond more. Through this, there will be a quite chance of controlling the conditions of Parkinson's syndrome. So, how dopamine is being deficiency? Dopamine deficiency, it's more of you can see here, mental health disorders, it's of depression, Alzheimer's diseases, Parkinson's diseases, mood swings, and in general, if we say about nowadays, due to the lack of nutrients, uh, diet, and all these, many of these will impact work pressure. All these started to impact the uh, human brain more and more, like complete workaholic guys also started to get some mood swings, and easily they do get uh, depressed, as if their work is was not done, and that leads to critical issues, like many guys are being also taking their lives in this aspect. So to control all these things, usually dopamine deficiency can be done. So how 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 dopamine works inside the brain system, as this brain reward systems helping to reinforce certain behavior that results as a surge of dopamine prompts into a laboratory pellet of a food or a product is being done. uh, the stress response, there are catecholamines, like dopamine. When it is being used, it can be used as dopamine receptors, or protein found in the brain and nerves throughout the body. If neurotransmitters are the nerves chemical messengers, then receptors are the nerve cells chemical receivers, as dopamine signal process can be done. uh, this helps in it plays an important role in many neurological processes, including movement coordinations, motor controls, conjugations, memories, and euphoric effects, pleasures, feelings of pleasure, certain drugs like cocaine. Dopamine creates especially certain feelings of pleasure. Certain drugs, such as like cocaine, can also cause a large amounts of dopamine to flood the system, producing euphoric effects which are very high, and that leaves the user wanting a more. So there are many drugs which are being available in the market, like Rifac, Neofib, Mirpax, Rampeloxine. Many of these are used for Parkinson's, insomnia diseases, and mental illness also. Hallucinations. The control of these disorders can be done by pathological gamblings and compulsively eatings, and dizziness can also be withstanded. These are the psychiatric conditions which are being studied and understood uh, through the impact of dopamine. So, what we we do is there are several methods like to detect this, to detect dopamine. How far this has been responding in a human cells. There are many different methods like capillary electrophoresis, colorimetry, liquid chromatography. Chromatography varies with various HPLC detection methods and calorimetric, calorimetric second variations. So native fluorescence detections have been used for reliable detections of dopamine. As I have, you can go through a paper which have been published in Journal of Electrochemical Society in 2018. How we have successfully done the determinations of dopamine using certain iron nanocomposites, and continued with graphene composites, and continued, and we also took forward with different types of zinc and iron composites. How metal ions are conjugated. When metal ions are conjugated with graphene, how it impacts the detection of dopamine, and also its simultaneous accelerations on other ascorbic acids or many other interfering compounds. So coming into this electrochemical analysis, electrochemicals we detection of dopamine was, although it has very disadvantages, like it's a very complex system, and like I said earlier, all these chromatography techniques, all these are high equipment, like highly costly, and it's a time consumption things, and labor, high number of educated labor is to be needed to analysis where it cannot be done. So to eradicate, means to minimize this disadvantages, we use electrochemical analysis, and through electrochemical sensors, electrochemical sensors, we use two or three main kind of electrodes in this, like basically glassy carbon electrodes or screen printed carbon paste electrodes. These SPC, screen printed carbon paste electrodes, are user friendly, like use and throw, one time use, so it anyone can be known the details and readings of the conditions. For electrochemical sensors, an non-modified electrode is capable for knowing the selectivity and the detecting ranges of dopamine, which is high in sluggish, and according to the how much of electrons are being transferred, and what would be the sensitivity between the material and the host electrolyte, and also about the inclusions what we are giving, if there is a poor selectivity, and any fouling effect is been happening or not, where the reproducibility can be done or cannot be done, or there any overlapping systems in between, if there are any overlapping, how to be overcome with that, and the over potential analysis or the potential static analysis, how to vary it, if there is a poor interference, how can it can be enhanced, all this was being studied. So basically, the trace level of electrochemical determinations of neurotransmitters, dopamines in biological samples. This was our major work, which we are doing from many ways, as I can say. Uh, Before saying like electrochemical analysis, as you can see in this image here, this is a reference electrode where in a three electrode systems, one counter electrode and working electrode, where our material is being coated onto the top of the surface, and instead of like like here in this, we are using glassy carbon electrode. If it is not glassy, as I said earlier, we can use SPC or carbon paste electrode or gold electrode, silver electrode, according to the material which we are using. It can be varied, and also working electrode derives through different variations. Like according to the even many researchers nowadays, there are many scientists, they do use nickel foams for biosensing applications directly. They are starting about using carbon cloths, carbon yarns, and PVTs, and directly using a polymer or a biomarker they are conjugating. And these things, as you can see in this image, it is quite clear how the analysis redox and oxidation potentials has been done, and how their readings are being analyzed in a simple manner. So, coming to this, using cyclic voltammetric studies, cyclic voltammetric studies is a phenomenon uh, to know how our uh, material is being covalent with the electrolyte and to the material which we have to the nanocomposite which we are prepared. Usually, cyclic voltammetry is various with different types of techniques. Like quite number of techniques are being used, like cyclic voltammetry analysis, differential pulse voltammetry, amperometric analysis, LSV, SWP, linear sweep, square wave voltammetries, and it varies on, and EIS systems, and basically they'll also been just targeted and checked over in ferricyanide systems also. So here in this, as you can see, uh, we have taken a metal nanocomposite. After using a metal composite, coating in a metal nanocomposite onto the surface of the glassy carbon electrode. For example, uh, when the dopamine is being added at an intervals like from 0.01 mole to 0.01 mole, like 1 nanomole to uh, 450 micromole. The variations, how the variations has been happen, uh, how high the current is being increased, and how high the dopamine and enhancement is being uh, happening in to the as prepared nanocomposites is being checked in this amperometric analysis here, to justify that, uh, as you can see here, the least amount of current which has been obtained and the highest amount of current which is obtained, we give a linearity range, like saying that the linearity explains about the performance of the sensor and its feasibility, whether it can be medically diagnosed or not, according to the conditions and to the pulse wind radiations. As we are going very journal today, we would like to say about, as I said earlier, we do these to be done in real samples, like as dopamine is being checked. Uh, the nanocomposites were brought in, and the nanocomposites were being checked through using a GC electrode in different samples, like blood serum, human blood serum, urine, whole urine, and synthesized urine, and sweat. Sorry, how the variations has happens, like when certain amount of material, if it is coated, what if dopamine is purged, like 50 microliter to 100 microliter, what would be the variance if the material is about 1:2 ratio, and dopamine is about 30 microliter to 200 microliter, what would be the ratio in the blood serum, and what would be the standard, like there is a standard protocol, like what would be the standard and the amount of detection which we can do, where how our material emphasizes, and how our material brings an analysis to this, saying that the material prepared nanocomposites has a perfect feasibility, can be, it can be studied through real sample analysis. As uh, you can see, this was one of our research group's paper, and we do continue our work. as for the time being, and I want to conclude here. So the future analysis, the future of this would be like to enhance the uh, living possibilities of humankind, like how to bring him out of stress, to bring him out of dizziness or mental illness or loneliness. Many pathological gambling should must be done, and it should be variations, and it should also help in controlling. So to control all these things, how dopamine can be used in a wide factor way, it, which is need to be studied, and how it pathologically inhabitates into the human body can also be studied, and that needs to be checked out. Many antipsychotic drugs are being available in the market, like dopamine androgens. Uh, how they're going to block the receptors in the brain. These analyses are also being done by many of the researchers in the world, like from Stanford, Yazzie group. They are and many of the professors, they are being professors and research scientists. They are working on how to enhance the welfare of the humankind. They're also been checking out, especially the future will be on more about deep brain simulations, where to help, especially the Parkinson diseases patients, and this would be the future working holes of this of neurotransmitters, especially through dopamine. And that's it for today. In this lecture, I'm gonna conclude, and this or that's my professor, PhD guide, and a renowned scholar who is a renowned scientist in electrochemical analysis throughout the world, Professor Shenming Chen, and my fellow lab mates and colleagues who works in electrochemical analysis and biological systems, biosensing applications, and sensor emphasizes the electrochemical derivatives. And that's it. I sincerely thank everyone for being a part of this. I sincerely thank everyone for giving me this opportunity. I thank all everyone, and we do encourage all sort of comments and questions, which is open to all, and deep conversations are also deep conversations, queries, any type of any kind of discussions are certainly welcome. And thanks a lot, and thanks everyone. Shisha is called thank you in Mandarin language, and thank you, thanks a lot. Bye bye. Okay.